So electron microscopy is this amazing tool that has developed and it has had many upgrades. So here we have 3 upgrades: time resolved, single particle, and cryo. We're gonna get into detail with that. So first of all, what is the purpose of microscopy? What is the goal of microscopy? We're trying to look at small things, yeah? Microscopy. We try to observe small things. Why? We want to know how things work. How do you know how things work? Well, you cannot just look at the parts. You need to look at the parts and how they interact, right? So knowing that you want to look at the parts and how they interact, what is the ultimate goal in biophysics? So we would like to have a continuous movie with 3D atomic resolution of the different molecules in a cell interacting in every possible way. So if we parameterize that, we would want temporal resolution, delta t, which is infinitesimal. This S with a dot is supposed to be a delta. So an infinitesimal time resolution. We want a spatial resolution, which is less than an angstrom. Then the different kinds of cells the different kind of molecules would be all of those in a cell. And then the observation would not perturb the system. Okay, so that would be a nice thing to have, but what we can achieve today is observing stages of the interaction in static images. We do have an impressive resolution, and we can do this with a couple of molecules. So our time resolution we achieve today is about 10 milliseconds with the technique that I'm going to talk about. We get a time resolution of about, we get a spatial resolution of about 2 angstroms, and we get 2 particles interacting, and then usually like you have ATP maybe to make the interaction happen. And then you destroy the sample because, well, you know the many ways to destroy your sample in electron microscopy. So in this talk, I'm going to talk a bit about the evolution of electron microscopy, how we get to this time-resolved single particle cryo-electron microscopy. I'm also going to talk about each of these things. For the time resolution, I'm going to refresh your mind in some chemistry that you probably saw in high school. And also I'm going to use the example of actin and myosin, these molecules that, these motor molecules that are what make us, that give us the ability of contracting our muscles. So starting with Ruska's electron microscopy, for which he got the Nobel Prize. Well, what does this mean for our understanding of muscles? Actin and myosin, we already knew that they, like since 150 years ago, something like that, that they are the responsibles of contraction, but the mechanism, the way this happens was unknown. In 1957, Huxley observed this longitudinal section of a muscle, and then he expected to later observe this structure here on the right. Yeah. And so he said that, yeah, it's probably actin and myosin. We just don't know the mechanism. Well, remember this image here on the right? That's exactly how we model muscles today. So this is, we have a thick filament in the middle, which is static. And then we have this actin filaments, which get pulled by the myosin, which is these heads here, which look like 2-headed things. Okay, so that's that was basic electron microscopy. Then cryo-electron microscopy was invented. We got another Nobel Prize for that. And so you mix your components and then you put them on a grid, then you freeze that. We already saw that in class. But the thing is, you mix your components and then you put them on a grid, okay? So remember that for later. So what does this mean for our understanding of actin and myosin? Well, these guys, they got the molecular structure of F-actin. F stands for filament. So actin is a small molecule. You get a lot of these small molecules together and you get F-actin. And then in yellow, you can see the binding sites, which they deduced also thanks to observations of with cryo-electron microscopy in year 1987. Okay, so as I said in the beginning, electron microscopy was a revolutionary technique, and then from that we have built new, well, we have upgraded it, yeah? So this is another upgrade, single particle. What does that mean? Well, imagine you have a sample and you obtain 10,000 of this molecule and facing towards you, yeah? So you get 10,000 different molecules like this one facing towards you. Molecules are the same, they should be. And then you rotate them to put them in the same position, then you average across all of them. And after many averages, you get like your average molecule. So after 4,000 something molecules that you superimpose, you get this nice molecule. This is the same as those phase averages. I don't know if you've seen these before. So this is like the average German phase. Of course, no one person looks like this, but this is what an average person looks like. So that's the same for these molecules. That's called a single particle, I guess, algorithm. It's not only for electron microscopy, of course. Then we get time resolved cryo-electron microscopy. So this paper here, you might see a familiar name. Well, it's a time resolved cryo-electron microscopic study of the dissociation of actomyosin induced by photolysis of photolabile nucleotides. And here we have the name of Dr. Schröder. So what he did in 1991 was he had this actin filament. They call it decorated with myosin. So it's an actin filament with lots of myosin in it. And it had ATP on it. And then they activate the ATP, which is, well, they fire away the reaction. They activate the reaction, then they freeze it. They activate this with light. That's why it's photolysis. They activate the reaction with light. They freeze it quickly, and then they observe what happens in the reaction, well, before the reaction, or 20 milliseconds after, or 30 milliseconds after, or 2 seconds after. So what you see here, and it took a while for me to actually see it. So here you have a rigid filament of actin, and then after 30 milliseconds of the start of the reaction, the filament looks wiggly, yeah? And it's funny because in the paper he says, we have to assume that this happens in all 3 dimensions. So they only have 2D images, yeah? This is back in 1991. And then after 2 seconds, we have a rigid filament, actin filament again. So this is the dissociation of myosin from actin. So you have myosin, which is sticking to the actin. You activate the ATP, myosin dissociates. In the process, actin gets like wiggly. And then when it is completely dissociated, actin is rigid again. And you can actually see you have these shapes, like this arrow shapes here. This is the myosin heads, or that part of the myosin. And then 2 seconds later, you don't have the myosin anymore, but you get the rigidity once again. Those sort of things you can do with time-resolved cryo-electron microscopy. And then in 2009, so this is quite recent, these guys created a new technique to make even better time-resolved cryo-electron microscopy, which is called mixing and spraying. So instead of mixing your sample or creating your sample and then putting it on your grid, you actually combine your 2 reactants and spray them quickly on your grid, which will then be plunged into ethane. and you can have different reaction channels, so you can have different times of reaction. And then the plunging it can take less than a millisecond, and then the freezing itself takes less than a millisecond. So if you do a very, very quick reaction and you plunge it very, very quickly, you can get to theoretically about a millisecond of temporal resolution. But in the paper that I read, they didn't need to do that, so they had not tried that, but they said it was possible to get sort of like millisecond time resolution. Yeah. So that's like the logo of Heidelberg University, the future since 2009. So you get a technique to activate your reaction or to start your reaction. You can also do this with light, like Schröder did in 1991. And then you do single particle. Yeah, so you detect your molecules which are in the same stage of the reaction, which are in the same 3D position. Yeah, you detect them and then you rotate them so you can superimpose them, and then you average through all of them, and then you get your resolved particle. So the temporal resolution, as I said, this one I'm sure it's about 10 milliseconds, and the spatial resolution you can get up to like 2 angstroms. I found a paper where they said that with cryo-electron microscopy they got to 1.7 angstroms resolution. So that's a future, and Schröder is quite optimistic about the future of these techniques to further complete the movie, the continuous movie of the reaction between or the interaction between myosin and actin. So this is a low resolution movie of actin and myosin interacting. Low resolution in both in that the resolution of the YouTube video is quite low, and that the size of the grain used for the molecules is big. So here you go. That's how myosin works. Then, so this is what he observed. Yeah, Schröder. You get the ATP, and then the ATP makes myosin detach from actin, and then ATP gets hydrolyzed, and myosin gets cocked again. And actually, that's just something curious. The two heads are independent. Yeah, so it's not like they walk like this. They are actually independent. So yes, this is the process of actin myosin interaction. X-ray diffraction is it's still widely used. Just you cannot always get crystals. The blue area here of the pie chart is where X-ray diffraction is mostly used to get new information. And the yellow part here is where you usually use cryo-electron microscopy. The parts in the yellow ellipses here, that's where Dr. Schröder says we will fill the gaps of the interactions with these new cryo-electron microscopy techniques. So one thing he told me to tell you was this part where you have the myosin attached to the actin, and then the movement happens. Yeah, that's called the power stroke. So that's what I don't know. You can ask him later about the power stroke. So time resolution, yeah. Reaction coordinate. You have H2 and O2, and we know that if you make H2 and O2 react, you're going to get H2O, which is water, and H2O is more stable than H2 and O2 together. Yeah. So you make them react, and you get this energy, which is called the Gibbs free energy. But whatever. Okay, so you need to activate the reaction. Yeah, you need to give the system a bit of energy before it goes into this H2O state. And actually here, when you get to the peak of this graph, you have a species which is not detectable, which is H2O2. So if we zoom in, yeah, what we see is that H2O2, there must be a little bit of H2O2, but since this, what do you call it? Potential
well?
What? Potential what?
Potential well? I'm not sure.
Yes, potential well. Yeah. Since this potential well is too shallow. Thank you. Yeah, there's going to be almost no species here. They're going to turn immediately into H2O or back into H2O2. So what do? So imagine you have a ball bouncing here. Yeah, that's the energy of your molecules. If you cool down your molecules, they're going to be bouncing here just a little bit, so they're never going to have the energy to turn into H2O. Yeah. If they are bouncing, if you give them much energy, then they're going to have the energy to bounce into the H2O area, and they're not going to have the energy to go back to H2O. So reactions take time. Yeah. In a given moment, your molecules are going to be in this H2O2 state. And if you then cool down your sample, you might freeze it into this H2O2 state. So you might have actually some molecules in this part, and then you could detect them. So let's say you have a more complex reaction, which is what we have in, well, proteins and macromolecules. So you have your reactants. Some milliseconds later, you already have your products. But you know that there is intermediaries, yeah, and that's what we are trying to observe. So that's yes, that's it. So in conclusion, electron microscopy is a great biophysics tool, which has also been greatly improved. The most modern types of electron microscopy are being used to observe intermediate states of biochemical reactions. For example, we talk about the power stroke, which we want to understand further, and time result single particle cryo-electron microscopy allows us to have a spatial resolution of about 2 angstroms and a temporal resolution of theoretically about 1 millisecond. Yeah. So yes, many papers. You can ask me about them. Thank you for your attention. Any questions?